Okay, so today we are going to talk about cell differentiation and a little bit of stem cells too, but mainly we'll focus on the process of differentiation during development. It's not changing. Okay. So, as you all know, we have more than 200 different cell types in our body. And all these cells, they originate from single cell, which is a fertilized egg. And these cell types, they emerge gradually during development through process which we call differentiation. And the underlying principle of cellular differentiation is basically establishment of differential gene expression. I hope by now all of you have an idea about differential gene expression and how it may be achieved. I would like you to again think about early embryonic development in flies, for example, where we had maternal genes which establish gradients along anterior posterior axis as well as dorsal ventral axis, and then the whole cascade of gene expression through gap genes, pair rule genes, segmentation genes, eventually the homeotic genes, which give rise to identity of different cell types, is established. So all this is basically a gradual emergence of establishing differential gene expression during early embryonic development. Now, in multicellular organisms, this diversity or diversification of cell types is extremely important, and each cell type has a characteristic cell type specific gene expression program. So if I say, an epithelial cell or a nerve cell, so nerve cell and epithelial cell, they both contain 46 chromosomes, but they are developing differently, not only morphologically, but biochemically, because both of them have to perform different functions. So they contain same set of genes residing on these 46 chromosomes, but they are both going to express different sets of key developmental genes, which are going to induce a different biochemistry here as compared to this one. And similarly, fat cell versus olfactory neuron, red blood cell, retinal cell. So all these are some of the few cell types we have in our body. Now they have to develop differently because each one of them are going to perform very specialized function. So it means when we say establishment of cell types, or we can also say specialization of cell types, specialization of cells. And this takes place during early development. They are developed, they are established during early development, and this specialization of cell types is due to the pattern formation. You already know how, so this is a mechanism of laying down a pattern during early development. how can we understand then changes in the gene expression? How can we understand differential gene expression? If you remember in our last recitation where we talked about tools, we did talk about some of the methodologies which can be used to characterize differential gene expression between different cell types. If we have, for example, a neuronal cell, neuron, and muscle cell, if we have to characterize what kind of genes are being expressed in each of this cell line, all we need to do is to isolate total RNA, because it will be at the RNA level we will learn what is being expressed in this cell type versus this cell type. And this RNA, we label them using different fluorophores. We, for example, this one gets Psi3 tag, and this one is tagged with Psi5. So muscle cell versus your neuron. And so they will be giving you different colors. You hybridize them with microarrays, or nowadays you can simply do, instead of, so this fluorophore labeling we used to do when we have to hybridize with microarray, and these are the microarrays. So microarray are basically glass slides, and on these glass slides we have a grid, you know, with a robot. We have each single dot represents a single gene, and you know, a couple of these glass slides, they cover all the genes in an organism, if you are working with mouse or humans or plants. And then you hybridize these ones with these microarray, and once you visualize, you will see, let's say Psi5 is the green one, Psi3 is the red one. So you will say, okay, all the green ones are the genes which are expressed in muscle cells. Red ones are the genes which are only expressed in the neuron, and the yellow is green and red, where the gene is expressed in both the cell types. This was done in, like, I would say maybe 10 years ago we were doing this, or a bit more than 10 years ago. But then came the next generation sequencing, where we simply take the total RNA from both cell types and we sequence each messenger RNA in neuron and muscle cells, and then we simply compare genes, how much are there. So here on the y-axis we have total number of reads, because if sequencing is going to sequence a gene A, let's say 10,000 times, it means you have 10,000 molecules of RNA in neuron total RNA, versus total RNA from muscles, let's say you have, you know, just 100 molecules being read. So next generation sequencing is even a step further than the microarray. It's a much, it increases our throughput, and you know, also genes which were lowly expressed, which are we are unable to detect here with microarray, they are sequenced in next generation sequencing. What are we achieving by doing this? By doing this, we are learning what in Wolpert says the luxury proteins. We are learning differential gene expression, or genes which are only expressed in neurons, genes which are only expressed in muscle cells. So the distinguishing features, the signature of neuron versus signature of muscle cells. Okay. And then once you have this, you can do wonderful experiments to prove, because this is not the end, this is just the beginning. Once you know the signature of neurons versus signature of muscle cells, let's say here you find 200 key genes which are neuron specific. Here you find 150 key genes which are muscle specific. Now you have to prove what is the function of each gene there and whether they are really associated with neuronal differentiation. So what you do, you take, for example, stem cells, and then you induce them, let's say with retinoic acid. If you add, you induce them to neuronal progenitor cell, which again, eventually gets differentiated into neurons. So let's say you do this in the cell culture, and you start depleting some of the genes here. So you took 200 genes, which you found on microarray or next generation sequencing. What do? You say, okay, I'm going to deplete out of these 200 gene A, and then go through this process and see if there is defect in differentiation. So you do this with all the genes one by one, and let's say you find, because depleting by RNAi, or nowadays you can use CRISPR for each gene, and then see if there are defects in neuronal differentiation, or the same thing can be done with the muscle differentiation as well. Once you find, let's say you find 50 genes really interfere with this differentiation from stem cell to neurons or to muscle cells, then you look at how they are connected. You develop gene network based on all the previous published literature, or you try to see if they are, how these genes are connected with one another, how they contribute to neuronal differentiation. At the end of the day, the purpose is, our objective is to find out how A to M to N different genes which we find here in these 50, how these genes are they different complexes? Are they, different independent biochemical complexes, gene networks which are playing a role, or they are connected with each other? So that's how we try to understand cellular function. Any questions so far?
Sir, can you explain this NPG? So what is this NPG?
Neuronal progenitor cells, NPC.
Okay, NPC, okay.
Before fully differentiated neurons emerge, stem cell goes to these cells, which eventually differentiate into.
And so when you mentioned that we are going to downregulate the expression of individual genes, so we are doing it like in one by one, or we are like, are we going to do it like if we find that there are 50 genes that are different in them, so can we do it like together or something like that? Can you explain that part as well, because I'm slightly confused about that bit.
So ideally, I prefer doing one by one. You can do pool as well. You can say, okay, I'm going to pool 5 double-stranded RNAs, and then see if they interfere with neuronal progenitor cells or the differentiation of neurons. And then out of, let's say you had nearly 30 pools, each pool comprising of 5 different double-strand RNAs of 5 different genes. You can then see which pool is really interfering with the differentiation. Then again, eventually you'll have to do single independent gene. individual genes.
Okay.
Clear?
Yes.
Any other questions? So remember, in case of flies, we saw this. We also discussed very briefly in vertebrate systems as well. The specification of cell types or determination of cell fates. So let's say we start our life here from single cell. And if we go back to our, so this is the stage in fly development where, you know, cell fate determination is taking place. So there is clear distinction between the time point where the cell fate determination takes place and eventually cell differentiation takes place. Already, if we remember the fly development, we saw, you know, the morphogen gradients being established by these early morphogens. Then we talked about the early signs of segmentation by coming from time of fertilization to the earliest signs of segmentation, where we saw just tiny grooves in the embryo, which showed 14 parasegments. Already cell fates are determined there at this stage, but they haven't yet differentiated. They may look still similar, cells may still look similar morphologically, but underneath biochemistry of each cell is different. If you remember the dorsal ventral axis formation, here at the ventral side, where dorsal protein goes within the nucleus and activates the twist snail, and on the dorsal side, you know, DPP mediated activation of genes takes place, where dorsal does not move into the nucleus here. So very early on in development, we see this pattern is being laid down due to differential gene expression, and fates of cells, you know, on the dorsal axis is being determined as compared to the ventral axis, and the underlying gene expression is different. However, if you look at morphology of these cells and the kinds of, you know, pattern, they may look same at the moment. But once cell fate is determined, now each cell, when it divides mitotically, so let's say this cell is going to make a neuron, and this one is going to make muscle. Now, gene expression is different, and each product of each mitosis, this arrow is representing mitosis. Each successive mitosis, which means 2 daughter cells here, then 4 daughter cells here. You keep going like this, and what you discover that the gene expression, which was established at the time of cell fate determination, which we say commitment to a specific cell lineage is taking place here. This is faithfully inherited during cell cycle, during all the way till differentiation, and after differentiation. You remember that, you know, I am a neuron, I have to express these key developmental genes to stay or to remain as neuron, and here a different sets as muscle cell. Eventually they start looking differently morphologically as well. So this process, when cell fate determination takes place, and, you know, key developmental genes are being expressed characteristically in a specific cell type. And this cell type specific gene expression pattern is then inherited through cell division to all the future progenitors. This is called cellular memory. And cellular memory involves transcription patterns. So it's maintained at the level of transcription. We also call this transcriptional cellular memory, or it's also referred to as epigenetic cellular memory. Okay. And I think when we were talking about homeotic genes in flies, we talked about Polycomb group and we talked about Trithorax group. So all the neuron-specific genes which are active in neurons and which maintain the cellular identity of neurons are maintained in the active state by Trithorax group proteins, and all the non-neuronal, let's say muscle-specific genes, they are switched off in the neuronal cells and they are maintained in the silent state by Polycomb group. And same here in muscles. The muscle specific are maintained by Trithorax in the on state, and neuronal specific here in muscle cells is maintained in the off state by Polycomb. Is it clear so far? And all this happens at the epigenetic level because each cell contains same 46 chromosomes, which means same sets of genes. So nothing is happening at the DNA level, but one level above. So at the chromatin level, these genes are being switched off or switched on. G, there's a question. I think Momina is there. G, Momina?
Yes, sir. Sir, I'm a little confused. So determination level, there's no expression of gene, right? No transcription.
There is expression of genes. So ये determination हो गई है. Dorsal moved into the nucleus and twist, snail activate हो गए. They activate their genes. Similarly, on the dorsal side, DPP activation took place, and dorsal could not move into the nucleus. So cell fates are being determined here, and then different genes are being expressed. But morphologically, they still look same.
So differentiation is when there's a morphological difference.
Yeah, differentiation is when they clearly start becoming different. They are part of different tissues as well now. For example, when you look at early embryonic development and, I give you another example here. You remember we had these pair rule genes. Okay, they all look same, but they are expressing different genes, we know. And at this stage, if you look at the cells, they all may look same, but they are becoming part of a different region in the body. They are not yet fully differentiated, but they are getting ready. They are fully determined. They are committed to cell lineage, let's say in this parasegment or this parasegment. They're never going to go to this parasegment now, and vice versa. They are never going to go to this parasegment. So they have committed to specific lineage, but morphologically, they still look same. And if you go slightly late at, let's say, 16 hours or 18 hours old embryo, or first instar larva, let's say in larvae, you will see these cells, they committed to different kinds of tissues. And this, when they start becoming part of specific tissues, this is a clear sign of differentiation. And eventually, when we have terminal differentiation, like now my eyes or my skin, this is a stage we call terminal differentiation. Now they have attained the ultimate status and they have even exit the cell cycle forever. This is terminal differentiation. Clear? Clear? Momna?
Yes, sir. So before terminal differentiation, can there be a factor that can change their commitment? Like, why are we separating the idea that there's a terminal differentiation and a part before that?
Yeah. So terminal differentiation is where there is no more differentiation. The process of differentiation is fully achieved and they have exit the cell cycle as well. Because each cell after differentiation, it has to exit the cell cycle. It no more divides. Because if it divides, then it's very dangerous. Let's say I have this skin cell, and if it starts dividing, it means it has to forget its differentiation status. It means it loses its cellular identity and enters into cell cycle again.
Okay.
A differentiated cell means extremely specialized cell type who is dedicated to perform a specialized function. And it no more divides. For example, a neuron. So this neuron, its function is to perform, sensory neuron or whatever kind of neuron is to, pass messages. Now, if this neuron forgets its memory, it means it's no more neuron, it will go something we call that de-differentiation. It will no longer look like nerve cell, it will be looking like some undifferentiated cell. And then based on what it enters into cell cycle and based on the signal it receives, it may differentiate into different cell type, muscle cell type. And that is required here at the place, and that may lead to cancer. So if I have this skin cell forgetting its epigenetic cell memory or transcriptional pattern, it means it loses its cell identity and it gets de-differentiated and then differentiate into, it tries to differentiate, but it's in the skin environment. It's getting all skin messages. So that's how the onset of cancer takes place. So cells after differentiation, at terminal differentiation, they have to exit the cell cycle. Did I answer your question?
Yes, sir.
Okay. So we have to understand what controls the cellular differentiation. So cellular differentiation is actually, it involves during early development when cell fate determination is taking place. If you remember human embryo, we had these earliest distinction was, these inner cell mass, the disc shape, all the cells which are going to contribute to future embryo. And then non-embryonic cells are these ones which we call trophectoderm or trophoblast cells, which contribute to placental development. They provide connection to our mothers. Now, these cells, inner cell mass cells, you remember we had this primitive streak emerging along the anterior posterior axis formation, and then through gastrulation we had these morphogenetic changes where embryo goes into three-dimensional structure, and we have ectoderm, mesoderm, and endoderm. So this is the first ever cell fate determination that we have three different distinct cell types. And then from ecto, further lineages are derived, you know, gut, endo goes into gut, and, you know, ecto makes skin, and different kinds of cell types are then established, or their fates are established. How all this is achieved? There are cellular signals. There are, you know, cell signaling events, and cell signaling plays a crucial role in early development based on what is the, so if this is now this embryo proper, you know, each cell is lying next to similar kind of cells, but based on the environment they are in, just like in fly, we had dorsal ventral axis and anterior posterior axis formation. Different cells, they start receiving different signals based on the environment around them. There are, and you know, these cell signaling, like paracrine factors, we learned about the paracrine factors. These are diffusible molecules which play a crucial role in the cell signaling events. Also, you know, the permissive interactions of a cell with the extracellular matrix proteins, that plays a role in the process of differentiation. And we have, when we talked about cell signaling, we talked about instructive cell signaling. And in case of instructive cell signaling, we said that cell has limited options. So signal A is going to get definitely a response X. A signal B is going to have a response Y. And all these changes, then such signaling ends up at the level of transcription. Cell type specific gene expression program slowly starts getting established. And then gradually it becomes specialized. It becomes more and more complex. Initially, it may be few genes, like in case of flies, dorsal moves into the nucleus and twist and snail comes in. Twist and snail are transcription factors. They upregulate 200 genes up and, you know, another 300 down. So these 200, 300, some of them will be further transcription factors. They will act, and gradually with time, this transcription pattern or cell type specific gene expression patterns, they become more and more complex and more specialized. Along the process of differentiation, in response to Momina's question, I talked about a cessation of proliferation, which means there has to be a time when cell division stops. So if we go back here, cell fate determination took place. Along the way, they became clearly differentiated, but then there comes a time where they stop dividing, no more proliferation. The reason is, if they proliferate, our morphological development will be abnormal. For example, if we divide all the cells which make our face, if all of them divide one more time, if they don't stop at right time, you know, our face will touch the floor. And similarly, if all the cells in our arms, they divide one more time, our hands will be touching the floor. So it means there is a tight correlation between cell fate determination, differentiation, and cell cycle. So differentiated cells, they know at what time to exit the cell cycle and attain the terminal differentiation. So everything on my face is in terminally differentiated state. So my ear lobes, my ear, eyes, nose, they are no more dividing. Okay. Well, that's a different question that our skin, it has, daily these cells die. When I shed my, when I go like this, millions of cells shed away from my body. They are dead, and they are replaced by newly terminally differentiated skin cells, which are being provided by the stem cell niche under the skin. So this is a top layer of skin, okay? And then underneath we have these stem cells. They keep dividing, they keep becoming specialized, and then these ones disappear, the old dead skin, and they are replaced by freshly differentiated cells. We did talk about nerve cells. They don't differentiate after they attain terminal differentiation, and same is the case for muscle cells. Now, what are the mechanisms for establishment, maintenance, and the inheritance of differentiation? So far, we talked about at the level of transcription. But what we are learning now in 21st century, that the control of gene expression, which contribute to the process of differentiation, is very complex. It indeed involves transcription, so switching on genes or switching off genes, but RNA splicing also plays a role, and then there are translational controls as well. So in case of RNA splicing, for example, we have a gene being transcribed, and it has exon 1, 2, 3, and 4. A differentiate specific. So this is, let's say, is expressed in stem cells, but it could be that instead of, so this is how the mature messenger RNA will look like, E2, 3, and 4, and then it's protein. But, it could be in a certain cell type, we have E1, 3, and 4, or E1, E2, E4, okay, or E2, E3, A4, or E2, A4, E2, E4. So it means from same gene, from same messenger RNA, we may get a different protein isoform, and they may be performing different functions. So besides transcription control, we have post-transcriptional control as well. We have non-coding RNAs. I would like each one of you to go and watch Professor Helge Grossan's. So you must watch the seminar by Helge Grossan's, which is on the YouTube. He appeared in our Zooming MCB session, and he talked about the clock. And one of the major discovery Helge led was a non-coding RNA LED7. In C. elegans, what happens from one larval stage to another developmental stage, if this non-coding RNA, which does not encode a single protein, it does not encode a single protein. It's a non-coding RNA. Rather, it goes and it blocks translation of other key developmental genes. And then development stops. This is a post-transcriptional control. So please go and watch Helge, Grozan's talk. Abdullah, can you share a link with them?
Okay, sir.
Okay, so this is very important. You should watch. And also, you should watch another talk, which was Susan Gasser. Both the talks are about development. So this one is C. elegans. And Helge also talks about C. elegans. These two talks are going to give you an idea about control of gene expression during development and also the methodology they are using. Watch completely. They are going to be part of your final exam, jo ke ye main midterm mein lunga. Okay. I'm not going to make question out of Susan's talk or Helge's talk. What I'm going to do is the question will be in such a way that you will have to adapt a strategy they are using. And we have discussed those strategies in this lecture as well. Then there is translational control. Translational control is like different isoforms of messenger RNA. So maybe you have same protein, no difference between the two proteins. But one of the protein becomes phosphorylated, the other one does not. Now, this protein could be functional and this protein could be non-functional. Okay. So now comes the third talk, which is Tony Hunter. हमने जो पेपर पढ़ने थे ना, ये इनकी टॉक्स में वो पेपर आपको बड़े अच्छे नजर आ जाएंगे सारे. लाजमी नहीं कि हम पेपर पढ़ें, ये पेपर डिस्क्राइब कर रहे हैं. जैसे आपने पेपर डिस्कस करने थे, ये लोग वो पेपर डिस्कस कर रहे होंगे. अब्दुल्ला, send them links for Helge, Susan, Gaza, and Tony Hunter's talks. So Tony's talk is going to show you the translational control. All three are, I would say, the most beautiful talks. Of my 21 years, I have attended many conferences, seminars, but they took really, because these were full-fledged research talks, an hour long, so they really took time and listened to question answers. So Tony talked about phosphorylation, how same protein becomes different, and now they are regulating different gene expression patterns. Okay. Now let's look at the transcription control. Okay. How transcription control is achieved. This from Wolpert's book in the differentiation chapter. This is a very nice example, simple to understand example. What he has shown that if you take elastase gene from mouse and a growth hormone gene from human, you have, this is a human growth hormone gene. It has its own very specialized cis-acting element, and elastase gene from mouse has its own cis-acting element, like, a tissue-specific enhancer element, a cis-acting element. Cis means it acts next, so it's not on a different chromosome or altogether far, far away. It's next to the gene. Okay. Now you engineer a construct where you take human growth hormone gene and you clone it together with elastase cis-acting element, elastase enhancer. You inject your construct elastase gene under, human growth hormone gene under elastase cis-acting element, and you inject in mouse embryo, and what you discover that now the human growth hormone gene is expressed specifically in pancreas. Elastase is a gene which expresses in the pancreas. So you see, very, very specific, this red one. Unfortunately, I have scribbled so much. Do you see the red dot here? In this circle?
Yes, sir.
This red one is pancreas specific gene expression. So this should give you an idea how cell type specific gene expression is established by cis-acting elements. Now you must be wondering, this cis-acting element must be present in every cell of mouse body, every cell, here, here, here. Why this cis-acting element is only activating mouse elastase gene here, or now the growth hormone gene here? Because the proteins which are going to come and bind here, they are only expressed in the pancreas. Now you ask the question, but the genes which encode these proteins, they are present in each cell of the mouse. This brings us to the next level, that okay, each of this has promoter region as well as some cis-acting elements which responds only to the signal in this particular region or cells during early development at the time of pattern formation, which activate these key genes to go and bind to this cis-acting element, activate this at the time of cell fate determination and or gradual process of differentiation. Now, did you understand how complex is even just one control of gene expression involved in differentiation? Try to imagine this, try to visualize this in your minds. Did you understand what I tried to explain you? Yes or no? Imagination complete नहीं हुई? Yes or no? बोल दिया करो यार.
Yes, sir.
Kind of.
So in this context, so there are two proteins, for example, crab binding proteins, which so they contain histone acetyltransferase, histone acid that causes acetylation of histones, which is involved in activation, and you know the growth hormones, as we know, each hormone is made by different cell types, and you know PET1 activation of growth hormones actually does involve this CBP crab interaction, and this is how the complex of proteins, so RNA-pol2 we learned in eukaryotes activates you know the transcription of genes. It's involved in synthesis of messenger RNA. But this is not enough. It requires tissue specific tissue-specific activators, or there will be tissue-specific repressors as well. It's not only just activating cell type specific genes, you have to repress other genes. For example, muscle specific genes in neurons are to be repressed. So they come into action wherever genes are to be repressed. You build up this, complex where Tata binding protein, transcription factors, etc., and RNA-Pol2, they will be blocked. They will be held in a pause state by repressor molecules, and repressors bind to very specific cis-acting elements, and they exert their repressive effect. And vice versa, the activators, they via co-activators, the blue ones, which serve as an intermediate between the basic transcription machinery and the activators, and eventually lead to activation of RNA synthesis. This is, how tissue-specific gene expression may be taking place or getting established. What are other signals? We talked about extracellular signaling or paracrine factors, etc. This is just a cartoon which shows, ligand, a cell signal is coming, which is perceived by the receptor. It binds, the receptor is getting dimerized, phosphorylation is involved. With this phosphorylation, this signal is transduced to other kinases, RAS, RAF, and then you see a phosphorylation mediated signal transduction event takes place where you have MAP kinase, then MAP kinase. It eventually enters into nucleus, and this phosphorylation mediated signal going like this, it ends up at the nucleus and activates specific genes. Imagine this signaling taking place early on during our development, while cell type specific gene expression patterns are to be established. We have steroid hormones as well, and steroid hormones, we have the, for example, glucocorticoid hormone, we have estrogen, we have progesterone, etc. These hormones are different than these paracrine factors. So what these steroid hormones do, they enter into the cell. There are specific receptors which are present in the cytoplasm. These receptors are held in an inactive state by specific protein. As soon as they bind ligand, which is the glucocorticoid, in this case, the hormone, the inactivating protein releases it. Now it's fully active, and hormone bound receptor moves into the nucleus from cytoplasm, binds to specific DNA sequences, which we call, let's say, GRE for glucocorticoid response elements, DNA sequence, okay, in the promoter region. And you will find a burst of gene expression, let's say 200 or 500 different genes are activated in response to glucocorticoid hormone. Same is the case by estrogen or progesterone. That's why, this may explain you why they're highly discouraged to take drugs, the steroids, because they provide a very fast changes in the gene expression, and these 500, 700 genes are are going to activate downstream, or they are going to act on downstream genes. So the whole metabolism and the whole body responds differently. But during our development, this is how all these different mechanisms are routinely being used to set up cell type specific gene expression pattern linked to differentiation. For example, retinoic acid is also a retinoic acid is also a nuclear hormone. And if you add retinoic acid, let's say in fly cells, which are undifferentiated, you add Ra, which is retinoic acid, they start differentiating into neurons. So you can imagine, and this kind of steroid hormone mediated gene activation, which is related to differentiation, it's taking place normally during our development, during the process of differentiation. For example, in Drosophila, ecdysone is a hormone which is involved at the larval stage when the metamorphosis is taking place. Now, what is time now? Oh, should we take 10 minutes? Let's take 10 minutes break, okay? So, as we talked about the establishment of cell type specific gene expression patterns, we also briefly touched upon the maintenance of these cell type specific gene expression patterns, because they are very important to maintain the differentiated state of cells. And once cell type specific gene expression patterns are established, these cell type specific gene expression patterns are eventually maintained generation after generation in the life of a cell, so that the terminally differentiated state or the prior differentiated state are fully maintained. Here, for example, you have positive feedback where it is shown that the muscle specific protein, which is called MyoD, it maintains its own expression. So MyoD protein is formed. So if this is the MyoD gene, the protein is formed, it goes, binds to its own promoter and maintains activation, whereas you have this MyoD also activating many other genes. Similarly, other example is the homeotic genes in fruit fly. How the transcription patterns which are established, cell type specific gene expression patterns which are established early during development at the stage of cell fate determination. So homeotic genes are the ones, or selector genes are the ones which then are responsible for maintaining as well as in the inheritance of cell type specific gene expression pattern. Here, for example, just a cartoon which shows you have a transcription factor, and this transcription factor goes and switches on A, B, C. It keeps gene D in the off state and E in the on state. Now, each of this protein, imagine, goes and activates or represses other genes, as well as they may activate, like A activates B, B activates C, and E represses this. So it can be a very complex web of gene network. So how these transcription factors may be working. So for example, this cartoon, another one, you have a protein product of gene A in the nucleus. It goes, binds to the cis-acting element in A itself, as well as in B and in C. In B, it switches it off. So B is kept off, C is maintained in the on state. And these proteins, as A is binding to the DNA elements, it means once these proteins are expressed, so A is going back to the nucleus, okay? And it keeps B in the off state, and C could be a cytoplasmic protein or whatever. So there is an auto feedback loop of A, just like the MyoD, as well as it also acts as a transacting factor. Now, transacting factor is one which is, transcribe it's a totally different gene, and then it goes and binds to the cis-acting elements of different genes. So this is a trans. So A is a trans-acting also as a trans-acting factor. Once these gene expression patterns are established, we have in the earlier part of the lecture, I said transcription is maintained at the epigenetic level. Now, what is epigenetic level? Would you have, we have these histone proteins which are wrapped around, 146 base pairs of DNA is wrapped around one histone octamer. So you have H2A, H2B, so 2 molecules each, H3, H4, 2 molecules each. So this is called histone octamer, and it's wrapped with 146 base pairs of DNA. This is called a single nucleosome, then the linker DNA, and then again 2 rounds of DNA, 146 base pair, next histone octamer, so which means the next nucleosome. So if we linearize histone protein, so this is the N term of histone, and this is the carboxyl terminal of histone. Similarly, you know the sequence of the DNA. So both the DNA as well as the protein component of chromatin, which are the histones, both of them can be chemically modified. They get covalently modified. For example, in case of DNA, what we have cytosine, it gets methylated. Whenever, so CG, so if this is the 5' and 3' orientation of the DNA, so cytosine in this context can be methylated. A methyl group is added on the 5th position of the cytosine. So here, in this methylation, whenever it is in the promoter region of the gene, which we can here, let's say if the promoter element here gets these cytosines in CG dinucleotide context, they get methylated, it correlates with silencing. Gene is silent, gene is repressed, gene is switched off. Similarly, the histone N-terminal tails, proteins are made up of amino acids. If I just focus on the N-terminal of, let's say, there are different amino acids, alanine, lysine, glycine, histidine, you just keep writing amino acids, lysine, etc. So it turned out that, different amino acids can be, for example, lysine can be methylated as well. Lysines can be sumoylated as well. Serines get phosphorylation. Threonines get phosphorylation. You have, ubiquitination. Lysines can also be ubiquitinated. So these chemical modifications in different contexts, so of the amino acid which is getting, for example, histone H3, if the lysine 4, if we start from N-terminal tail, the lysine 4 gets trimethylated, it correlates with active genes. Similarly, lysines at position 9 or lysine position 14, position 23, 9 can be acetylated, 14 can be acetylated, 23 can be acetylated, 27 can be acetylated. So acetylate or histone H4, lysine 5, lysine 8, lysine 12, lysine 16, they all can be acetylated. So acetylation correlates with gene activation. Gene is on. So you remember the euchromatin and the heterochromatin. These are the two large classifications of chromatin at the level of structure. Heterochromatin is where all the genes are constitutively off, and euchromatin is where genes are on. This was in terms of, the structural chromatin structure and the gene activity correlation. But when we talk in terms of differentiation, there are many genes in euchromatin which are repressed, which are silent. It doesn't mean they're heterochromatin. Heterochromatin is a very different structure in the chromosomes. But many genes in euchromatin which need to be repressed during the process of differentiation, they may be kept silent through one of these mechanisms, DNA methylation, or here I said K9 can be methylated. So it can be, K9 can be methylated as well, but they are exclusive. K9 will be either methylated or acetylated. If it is methylated, gene is silent. If it is acetylated, gene is on. Okay, so this is how we, during process of differentiation, genes are switched on and switched off. Here, this cartoon is showing example of X inactivation. In human females, one of the two X chromosomes become inactive during very early developmental stage. Initially, both the X chromosomes are relaxed, open circular, genes are on, but then there comes a stage where one of the X becomes totally jumbled up, highly condensed, highly compact. It's visible under microscopes like this. We call this bar body, BARR, after the name of the scientist who discovered this. The other X remains fully active. And that's how when each cell where the X is inactive, it will mitotically divide. The one which is active will remain active. The one which is silent with or completely inactive will remain inactive. This is also very important during our development, because if both the X chromosomes are fully active in human females, the gene dosage becomes unequal as compared to males where only one of the X is performing function. So the cells, they die and abnormalities develop. Now, I also give you example of so when you have via DNA methylation, histone modifications, cell type specific gene expression patterns established early during development, they need to be maintained now through mitosis, like here in case of X chromosome, or let's say eye cells or muscle cells or whatever, neuron cells. This function is performed by two groups of genes which are evolutionarily conserved. And these evolutionarily conserved groups of proteins are called Polycomb group. We talked about this in detail when we were talking about in context of homeotic genes. Polycomb group genes are going to maintain cell type specific genes in the off state, and even during mitosis through cell cycle to the progenitor cells, they will be passed on in the off state, and the gene which is on cell type specific gene, which is crucial for differentiation, will be maintained in the on state by Trithorax group genes. And how they maintain this, so the Polycomb group, for example, I just write down names of, so there's a protein complex which is called PRC2, Polycomb repressive complex 2, and there's a protein called enhancer of zest in PRC2. It methylates lysine 27, gets trimethylated, histone H3, and this is bound by another protein complex of Polycomb proteins, which is called PRC1, Polycomb repression complex. One of the protein here called Polycomb, it recognizes this methylated state, and all the PRC1 complex comes and binds there. They maintain this here in the silent state, and after mitosis in the daughter cells, this phenomenon is faithfully inherited. We call this epigenetic inheritance. So we are not only inheriting during cell divisions the 46 chromosomes, but the chromatin structure is also being inherited through the local modifications of chromatin, histone modifications, etc. In mammals, the H3 lysine 27 methylation is known to direct the promoter methylation. So let's say the DNA, the nucleosomes. So if this N-terminal tail contains lysine 27 trimethylation, this can direct the DNA. So this is the DNA, for example, promoter. So the CPG sequences here will get DNA methylation and gene is kept repressed. Now, if gene is to be maintained in the on state, the Polycomb complex cannot catalyze this lysine 27 methylation. Rather, the Trithorax group is going to catalyze K27 not acetylation is just a single molecule. So if you look at the amino acid structure of lysine, you will see chemically only it's possible to add one acetyl group. So lysine 27, instead of getting methylated, Trithorax group acetylates that, and that is then associated with maintaining gene in the on state, and this is also inherited epigenetically. And this is how the selected genes, homeotic genes, they give not only identity to specific cell types, but even after differentiation, Polycomb and Trithorax then maintain the characteristic gene expression pattern of homeotic genes in cell type specific manner, because these selector genes, they are involved in giving specific identity. But then someone has to maintain those cell type specific homeotic gene or Hox gene expression pattern, and that's where Polycomb Trithorax comes in. So I think how much time is left?
We have 10 minutes.
So we will stop it here, this lecture, and after the midterm, Dr. Shoaib is going to teach you the Dev Bio, and Shoaib is going to start with the models of cell differentiation, blood cell differentiation, etc. Okay. Tomorrow at 9 in the morning, the seminar by Professor Dr. Steve Henikoff. It's a coincidence, Steve Henikoff. It's a coincidence that we are just talking about this epigenetic inheritance and, you know, the chromatin modifications and, you know, cell type specific gene expression patterns. So Steve is actually an authority in this field. And, you know, what I have covered today, you will hear this. So don't miss this seminar. Don't just come for that there will be attendance. I don't want you to be there just for attendance. I want you to learn because this seminar by chance is coinciding with our lecture, which is ending at the maintenance of gene expression pattern, or you know, how transcription gene expression patterns are inherited. Any question?
Sir, some of us have class at 9 a.m.
Which class you have?
I've got philosophy of education.
No problem. So you should watch this, because all the exams I'm going to do this before the midterm. So we'll have nearly 2 extensive, very difficult quiz exams before actual midterm. So that this Steve's talk and the other talks I said, they are on the SBSSE webpage, as well as Abdullah will share the YouTube link. So go there and watch. Okay.
Yes, sir.
All the others who do not have class, they don't miss the seminar tomorrow. It's must. I see you tomorrow morning, 9 sharp. Okay.
Sir, I have questions
Ji?
regarding So can you please explain the pattern? Like we will have a quiz this Thursday or a practice exam? What I didn't understand earlier what you were saying.
No, this Thursday we will have. You remember, due to death of my colleague's mother, we could not have our recitation. So this Thursday we will have our recitation in the lecture. You remember I wanted to cover last part of the tools. So on Thursday we will cover that, and then next week Tuesday we will have first exam, then the Thursday we'll have the second one, and we can keep going. You have to decide the date for your exam. Okay, finalize the date of exam. Talk to Ahmad whenever you want, and then we have enough time during March. Clear?
So we'll have 2 quizzes next week.
At least 2.
Okay.
And there can be 3 as well.
And sir, for all of the quizzes, the syllabus is everything we have covered till now, or
Yeah,
it's
everything. It's everything. And these quizzes will be an hour long. They will be like a mock midterm exam. Okay?
Sir, can we get a practice question or something?
No.
So we know right?
प्रैक्टिस क्वेश्चन मैंने करवाए थे अंदर लेक्चर के अंदर, याद है?
जी सर. तो उस तरह के होंगे?
उसी स्टाइल पे आएंगे.
ठीक है. All right.
चले. Any other question? So if there are no more questions, don't miss seminar tomorrow. I look forward to see you all. Allah Hafiz.